For the first time, an international team led by researchers from the Max Planck Institute for Evolutionary Anthropology have managed to sequence multiple individuals from a remote Neanderthal community in Siberia. Among these 13 individuals, the researchers identified multiple related individuals, among these a father and his teenage daughter. The researchers were also able to use the 13 genomes to provide a glimpse into the social organization of a Neanderthal community. They appear to have been a small group of close relatives, consisting of 10 to 20 members, and communities were primarily connected through female migration. The first Neanderthal draft genome was published in 2010. Since then, researchers from the Max Planck Institute for Evolutionary Anthropology have sequenced a further 18 genomes from 14 different archaeological sites throughout Eurasia to explore the social structure of Neanderthals. The researchers turned their attention to southern Siberia, a region that has previously been very fruitful for ancient DNA research, including the discovery of Denisovan hominin remains at the famous Denisova cave. From work done at that site, we know that Neanderthals and Denisovan were present in this region over hundreds of thousands of years, and that Neanderthals and Denisovan have interacted with each other, as the finding of a child with a Denisovan father and a Neanderthal mother has shown. In their new study, the researchers focused on the Neanderthal remains in Shagurskaya and Okladnikov caves, which are within 100 kilometers of Denisova cave. Neanderthals briefly occupied these sites around 54,000 years ago, and multiple potentially contemporaneous Neanderthal remains had been recovered from their deposits. The researchers successfully retrieved DNA from 17 Neanderthal remains, the largest number of Neanderthal remains ever sequenced in a single study. Shagurskaya cave has been excavated over the last 14 years by researchers from the Institute of Archaeology and Ethnography, Russian Academy of Sciences. Besides several 100,000 stone tools and animal bones, they also recovered more than 80 bone and tooth fragments of Neanderthals, one of the largest assemblages of these fossil humans not only in the region but also in the world. The Neanderthals at Shagurskaya and Okladnikov hunted ibex, horses, bison and other animals that migrated through the river valleys that the caves overlook. They collected raw materials for their stone tools dozens of kilometers away. And the occurrence of the same raw material at both Shagurskaya and Okladnikov caves also supports the genetic data that the groups inhabiting these localities were closely linked. Previous studies of a fossil toe from Denisova cave showed that Neanderthals inhabited the Altai Mountains considerably earlier as well, around 120,000 years ago. Genetic data shows, though, that the Neanderthals from Shagurskaya and Okladnikov caves are not descendants of these earlier groups, but are closer related to European Neanderthals. This is also supported by the archaeological material. The stone tools from Shagurskaya cave are most similar to the so-called Mycoquean culture known from Germany and Eastern Europe. The 17 remains came from 13 Neanderthal individuals, 7 men and 6 women, of which 8 were adults and 5 were children and young adolescents. In their mitochondrial DNA, the researchers found several so-called heteroplasmies that were shared between individuals. Heteroplasmies are a special kind of genetic variant that only persists for a small number of generations. Among these remains were those of a Neanderthal father and his teenage daughter. The researchers also found a pair of second-degree relatives, a young boy and an adult female, perhaps a cousin, aunt or grandmother. The combination of heteroplasmies and related individuals strongly suggests that the Neanderthals in Shagurskaya cave must have lived and died at around the same time. Another striking finding is the extremely low genetic diversity within this Neanderthal community, consistent with a group size of 10 to 20 individuals. This is much lower than those recorded for any ancient or present-day human community, and is more similar to the group sizes of endangered species at the verge of extinction. However, Neanderthals didn't live in completely isolated communities. By comparing the genetic diversity on the Y chromosome, which is inherited father to son, with the mitochondrial DNA diversity, which is inherited from mothers, the researchers could answer the question: was it the men or the women who moved between communities? They found that the mitochondrial genetic diversity was much higher than the Y chromosome diversity. which suggests that these Neanderthal communities were primarily linked by female migration. Despite the proximity to Denisova Cave, these migrations do not appear to have involved Denisovan. The researchers found no evidence of Denisovan gene flow in the Shagurskaya Neanderthals in the last 20,000 years before these individuals lived.